Hey guys, I'm Dr. Rashid Hashmi and you are watching Dr. Digital College. You know that in Edexcel International A-Levels Biology Unit 2, topic 3.19, you have studied that what's the structure of the DNA and how is the DNA working. And then there is one topic, epigenetic regulation of the gene expression. Okay, one of the epigenetic regulation of the DNA regulation or the gene's expression is processing of RNA, or you could say RNA splicing. In prokaryotes and eukaryotes, there is one difference that in prokaryotes, RNA is formed and it's readily used for the synthesis of some proteins. While in case of eukaryotes, RNA can be formed, it can be used readily just after its formation, or it can be stored for a long time and it is stored in inert form that it's not being used, and whenever it is used then it is processed. Okay, and for example in red blood cells there is nucleus, and when nucleus is given out as an adaptive feature for the working of red blood cells, and even then red blood cells can survive for up to 120 days. How are they surviving for 120 days? Means there is RNA present, which is controlling the synthesis of different proteins and all the functions of the cell. Okay, so let's see in this topic that how is this processing of RNA or RNA splicing brought about. It's there in all eukaryotes, and in some of the prokaryotes it's also done. In majority of the prokaryotes, this is not there. Now let's see that how is this done. Okay.
Today we are going to discuss about post-transcriptional processing of RNA. The word post-transcriptional means that transcription has occurred, RNA is formed, and after the RNA formation there is some processing done. You know that one of the major difference of prokaryotes and eukaryotes, although it's not in all prokaryotes and eukaryotes, this is present in few prokaryotes, but in all eukaryotes, that RNA is processed and it can be stored. It can be done in all eukaryotes, but in very few prokaryotes. For example, you know that we have red blood cells. Their age is about 120 days and they have no nucleus. What does it mean? That could they survive without any nucleus for 120 days, about 4 months? No, actually their mRNA is present in them. All the mRNA, messenger RNA required for the life of 120 days is synthesized from the DNA and is present, although there is no DNA. As there is no nucleus, so there is no DNA. So in a way you can say that command for control is present. If commander is absent. If commander is not there, but the command is present, that what different proteins are to be synthesized and what different proteins are not to be synthesized. Okay, now this is called epigenetic modification. this is epigenetic modification of RNA, and this is a kind of gene expression, that whether gene has been transcribed into RNA, the final product may be formed or may not be formed. Okay, so we can say that this is here DNA. For example, this is the DNA. From here to here, this is mRNA formed. Now this mRNA can be stored, and after storage it can be processed, and after processing a protein can be formed. Okay, so this is a lengthy process, and I told that this process is present in eukaryotes. This process is very rarely present in prokaryotes. Okay, but you know that eukaryotes, they are considered as the modern cells, means they are evolutionary more advanced cells, and eukaryotes, these are called the old cells or primitive cells, or you can say evolutionary less advanced cells. So eukaryotes should be at advantage, but apparently you can see that in prokaryotes, from DNA RNA is formed and then it is ready to be used. So I can write here that in case of prokaryotes, from DNA mRNA is formed, and mRNA is directly used for the formation of proteins. That is there in prokaryotes. There is no storage. This is readily used. So apparently this is the advantage, but you know that if we think deeply, this is a disadvantage. What is the disadvantage? That there is always a need of DNA, or you could say the gene. So some cells without DNA, without nucleus, they cannot survive. But in eukaryotes, such as I have just now quoted the example of red blood cell, they can survive without nucleus. So this is their special adaptation to accommodate a lot of hemoglobin and oxygen. They have excluded their nucleus as their adaptive feature for their function, but they can survive for up to 120 days. Okay. Moreover, there is another reason is that if there is one gene, it will form one mRNA, and from one mRNA there will be one product in case of prokaryotes. But in case of eukaryotes, if say for example there is one gene, they will form one mRNA, and now this one mRNA can be processed one way or the other way. Process 1, this is process 2. In process 1 there will be the formation of one product 1, and after the end of processing 2 there will be product 2. So alternative processing can give you more than one product. So we can say the total number of products which can be encoded by the genes are more than the total number of genes. So in a way, in eukaryotes there is greater utility of genes. Genes are utilized in a greater way, in a more versatile way. So we can quote one example of this one. Say for example, there is calcitonin and CGRP. This is a hormone, and this is one protein. They are encoded by same gene, but they are performing different functions. I'll be telling you a bit detail of it, but before this, let's go towards this RNA processing. that what actually this RNA processing is. RNA processing can be there for messenger RNA, it can be for tRNA, it can be for ribosomal RNA, or it can be for many other different types of RNAs. But for the time being, I'm talking about mRNA, okay, which is directly giving you the code of protein synthesis. Now, you know that if this is here your readily synthesized RNA, I can write it that this is crude mRNA. Okay. Now, within this crude RNA, there are some areas, just for example, I am adding here just two, which are called introns, INTRONS, introns. This is intron 1, this is intron 2, and the other regions, they are called exons. Let me write this one. This is here exon, this is here exon, this is here exon. So exon 1, exon 2, exon 3, different exons are there in it. Okay, so introns can be called as intervening sequences, or you could say they are interrupting sequences. They are cut and removed. Some genes may not have any introns, while other genes may have 1, 2 or many introns. There is a gene called LTBP2. This is related with human eye diseases. It has many introns. You could say several tens of introns are there in it. Okay. Now, when RNA is synthesized and it's in crude form, it can be stored. If it is not to be used readily, then it can be stored. Before to go towards RNA processing, let me tell you that what is the storage of RNA. Say for example, this is the RNA. On its two ends, there are some inert structures are added like this. There is one cap is added over here, and there is one tail is added over here. Now, what are these caps and tails? This is called methylation, and this is called adenylation. Basically, this is a cap of many copies of methylated adenosine monophosphates. So this is ATP, which is methylated ATP. Many copies are there and they are forming one cap. Similarly, what is this adenylation? This is a tail of polyadenylate. These are actually many copies of ATP. Okay, so when these two things are there, they are making RNA inert. So this is an inert RNA, means this RNA will not be translated. It will not be making some copy of protein. Okay, so in case of eukaryotes, RNAs can be stored by capping and tailing them by methylation and polyadenylation. They can be stored. So once they are to be used, they can be used just after their formation, by after processing, or after storage. When they are to be used, they are processed. when processing is to be done, their introns are cut and removed. Okay, so how is that done? Now if look at this, this is here RNA, this is one intron, this is one intron. There is a complex of enzyme called spliceosome. It will come and will cut and remove it. You could say just like endonuclease, this will cut and remove it. Mean how will remove it? This will loop out, this intron will loop out, and here it will be cut with the help of endonuclease. So that this is here your final RNA and these are the introns removed. These are cut and removed introns, and this is your final RNA. Now whole of this process is very important for the proper expression of the gene. This is called the terminology for this processing of RNA is called RNA splicing. Okay, and in this case there are specific sites on the RNA. Say for example, if this is here 3' and this is here 5' from the structure of the DNA, you can easily know that what is meant by 3' and what is meant by the 5'. Mean the sugars are there in such a way that their carbon number 3 is on this direction and their carbon number 5 is on this direction. So on RNA there are two sites. These are called splice donor site. The splice donor site is ending at GT towards 3' of intron. Okay, and there is splice acceptor site. This is ending at AG, sorry, not AT, this is AG. This is towards 5' of intron. Okay, what is meant by this splice donor and splice acceptor site? Say for example, this is the 3' end, this will be here GT and this will be here AG. Now AG will join with GT. This is, say for example, this is exon 1 and this is exon 2. Now AG of exon 1 will join with the GT of exon 2. So AG is splice acceptor site and GT is splice donor site. So this will join with this one. So from in between, this intron will be removed. So if there is any change by mutation in splice donor and splice acceptor sites of the gene and then of the RNA, then this RNA splicing or RNA processing can be altered, or this can, and then it can result in wrong expression or no expression of some gene. Mean the RNA will not be formed proper, and then it will not either be expressed, it will not be translated, or it will be translated wrong. Okay, so this is ultra simple way. There are many further details of RNA splicing. It's a full-fledged complete topic which you will study in your advanced classes. but here for simplification I have told that introns are cut and removed and the exons are joined to form the final RNA. Now if we go back with our example, I said that in eukaryotes one gene can form one crude messenger RNA which can be processed different way to form different products such as calcitonin and calcitonin gene related protein. Now let's see what are these two examples. Okay. Now if this is here one gene and it has formed one mRNA, and one mRNA is spliced or processed one way and it has formed calcitonin. Calcitonin, you know that this is a hormone which is produced by thyroid gland and it lowers blood calcium level. So it is regulating blood calcium level. So this is the job of this hormone and this is formed by one RNA. Now if this RNA is processed by another way, then it will be producing a polypeptide which is called CGRP. This stands for calcitonin gene related protein. Calcitonin gene related protein this is, and you know that this its function is vasodilation, that it will cause the dilation of blood vessels, and it is also produced in nervous system, central nervous system, brain spinal cord, and the peripheral system, different nerves, and its function is the transmission of pain stimuli, or you could say stress stimuli. So I can write here that this one is for CGRP is for nociception. This is called, the word is nociception, that is noxious stimuli are transmitted. Noxious stimuli means those stimuli which can cause trouble, they can cause pain. So through the sensory neurons, such stimuli, the noxious stimuli, they are transmitted with the help of this polypeptide, the protein CGRP. This belongs to the family of calcitonin. So this is, you know, that one protein, this is one function which is entirely different. This is function 1 and this is here function 2, which is done by a different protein, a different function. So we have one gene, one crude RNA, but it is alternatively spliced and different products are formed which are performing different functions. So in a way I can say here that there is greater utility of genes in eukaryotes as compared to prokaryotes. There is greater utility of the genes in prokaryotes. So I hope that this is understood to you, that how is RNA processing or RNA splicing done, what's its objective, what's its process, and overall what is it. Okay.
Thank you for watching this video, and hope you have liked it. That what's the processing of RNA in prokaryotes and eukaryotes. What's RNA splicing? Okay. Thank you.